The second example for which the linkage study was done by Morgan included the linkage in an organism, that is Drosophila. Drosophila is also called as fruit fly. The species which Morgan chose for understanding the linkage concept was Drosophila melanogaster. This was the species in which the study of linkage was done. In this, Morgan carried out various dihybrid cross experiments in Drosophila to study the genes that were sex-linked. These crosses were similar to the hybrid crosses that were carried out in the Mendel's experiment. Here, the cross was performed between the hybridized yellow-colored body and white-eyed females. That was crossed with the brown-bodied and red-eyed females. That is, this yellow body and white-eyed Drosophila was of female and that of brown body and red-eyed was of male. So here was the cross or the dihybrid cross that was performed between the two different sexes of Drosophila, where he observed that the two genes did not segregate independently of each other and the F2 ratio that was obtained was significantly different from that of the Mendel's theory. So, on the basis of this experiment, the logical explanation of linkage and Drosophila recombination was given by Morgan through his experiment. Let us study that why this Drosophila was chosen as for the experimental base. Now, Drosophila was chosen for the experiment because of the following reasons: that it can be easily grown in the laboratory, secondly, it produces large number of offspring, where male and female both flies are separate, and therefore the chances of self-fertilization is not possible. That is, the chances for self-fertilization cannot occur. And the lifespan of Drosophila was of about 15 days. So these were the reasons for which Morgan chose Drosophila as his experimental organism. This figure depicts the male and female fruit fly along with their type of chromosomes. As said earlier, he performed a dihybrid cross experiment between the grey-bodied color and long-winged with the black body color and vestigial wing. Out of these, the grey and long-winged Drosophila was considered as dominant, and this black body plus vestigial wing was considered as recessive. So these were the two parents with different characteristics that was chosen by Morgan to perform a dihybrid cross experiment. Now, on the basis of this experiment, as said earlier, the F1 generation was expected according to the Mendel's theory, where the results of the F1 generation should be in manner that the fruit fly that are obtained is grey-bodied and long-winged. But the difference that was obtained was in the F2 generation. Where the result was in the form of 50% grey bodied and long winged, while the rest 50% were black and vestigial winged. Thus, no flies were obtained with the new combination of characters. That is, although in F2 generation the result obtained was totally different from that of Mendel's theory, but no new colored flies were obtained. So, the such results that were obtained indicates complete linkage. And the cross that was done to obtain F2 generation was between the F1 flies with the parent that is recessive for both the characters. That is, we can say that for F2 generation, a test cross was performed between the flies, which resulted into this containing 50% of both dominant and recessive, on the basis of which it was also proved that no crossing over occurs between the flies, that indicated the complete linkage. This figure depicts the complete linkage within the Drosophila, where two different character containing parents are chosen, that is grey and long winged, which is dominant, and black and vestigial winged, which is recessive. The F1 generation obtained was expected, but within F2 generation, where the test cross was made between the F1 and the parent with recessive character, the result obtained was 50% of both dominant and recessive character containing parents, which indicate complete linkage and therefore no crossing over occurred between them. But after obtaining the F1 flies, he carried out another experiment where he used the F1 flies that was crossed with the parent containing double recessive male flies. In which the F2 generation obtained was totally different. That is, the parental combination that was obtained was about 83%, while the recombination of such a cross that was obtained was 17%. That is, the parent with grey, long winged was about 41.5%. Then the parent containing grey and vestigial wings was 8.5%. The one with black and long-winged was again 8.5%, and the last, that is black and vestigial winged, was 41.5%. That is, these two, the grey and the black vestigial wing, was constituting the 83%, which was very obvious. But the two, that is grey vestigial and black long wing, showed the recombination process where the crossing over occurred between the two gametes that resulted the 17% of one. This figure depicts the second experimental result where the linkage and crossing over, that is recombination, occurred. As it can be seen from the figure, the parental combination, that is black and vestigial wing and grey and long wing, was 41.5% each, that constituted the 83% of parental combination. But the second, that is crossing over or recombination between the two flies, that is black and long wing and vestigial and grey wing flies, constituted 8.5% each, that is the crossing over or recombination between the flies was about 17%. However, Morgan explained this unusual ratio because it was of the presence of the gene for black and vestigial winged that was present on the same chromosome. That is, the gene that was responsible for the black body color and vestigial wing was located on the same chromosome. And that was the reason why such a recombination occurred, according to Morgan. As a result of which, these genes were linked together. That is, linked genes occur which caused the linkage type of inheritance. The two pairs of genes that were obtained in the F1 generation, that is G and L, that is the black and grey body colored, while the long and vestigial winged parent were not considered to be assorted independently. Thus, in a dihybrid cross, the Mendel's law of independent assortment was not completely true, and therefore, the Morgan theory of linkage and recombination proved to be true. This figure depicts the crossing over process in the Drosophila fly, where the centromere of two different chromosomes that occurred on the pair got exchanged, and as a result of which the recombination occurred. On this basis, recombination was defined as the crossing over of genes that leads to the generation of a non-parental gene combination. Thus, on this basis, the physical association or the linkage of the two genes described the term linkage with the help of recombination or crossing over process. And thus, the physical association of the genes on chromosome was also proved. This figure depicts the recombination process in any organism where the cell, after undergoing meiosis, exhibits crossing over in the recombination process, and thus two non-parental gene combinations are formed due to crossing over of genes. That results into a new gene combination and its formation.